Welcome to AFB Teachings. In last class, we have already discussed about what is nutrition and what are the type of nutritions in plants and animals. So we learned already autotrophic nutrition in plants, autotrophic nutrition and heterotrophic nutrition. These are the two types of nutrition. So autotrophic means in green plants, heterotrophic in animals, and then we already discussed about this photosynthesis. So these green plants, how the green plants prepare their food in their bodies itself. That synthesis is called photosynthesis. In photosynthesis, what happens? Four factors. In last class already we discussed about these four factors or four conditions needed for photosynthesis in green plants. So first factor is sunlight. Second one, carbon dioxide. These are from atmosphere. And third one, water. Fourth one, chlorophyll. These two in plants. These two are present in already green plants. Water is present in green plants. Chlorophyll is present in green plants. And they should take sunlight from the atmosphere and carbon dioxide also from the atmosphere. And then these four factors, we call them factors for photosynthesis, are the conditions, main conditions for the photosynthesis. So these are the four factors are needed to undergo photosynthesis in green plants. Now, where does it occur? That means in plants, in green plants, we are saying that in green plants, this photosynthesis undergoes or occurs in the green plants. If we draw a plant like this, then if we have a green leaf, we should draw a green leaf. Then where do the photosynthesis? In the leaf means where? In the leaf means in the midrib or outer layer of this leaf or in the middle. Like that, many questions are there for where the photosynthesis occurs. So in 8th class already, we have learned that every living organism have cells in their bodies. So life, which have life, the animals or plants which have life have cells. That means all body is filled with cells. So cells are in 8th class, the second lesson name is also cell. The what do you call it this? Cell is the life for the animals and plants. Unit of life. Cells are the units of the life. If we go for non-living, what you call in non-living, that means like this board, in this pan, this wall, this cloth have no cells. But in plants and animals, these bodies have cells. Plants body have number of cells, that means crores of cells in each their parts. And animals also, if we check our eye or ear or nose, we have crores of cells in our eye. Like that, in body, crores of we have to make it multiplication. So cells are the unit of life. If there is any cell, that means it is a living thing. And non-living have no cells. This is the most important thing we have to learn. Already we have learned in the 8th class. Cell is the unit of the life. So plants have already cells. So what we are talking, photosynthesis occurs. Where does it occur? We are telling that chlorophyll is present in the leaves. So in the leaf, the photosynthesis undergoes or occurs in the in the leaves itself. So in leaf means in 8th class already we learned now. So these leaf have cells. If we take animal, that means our body also having cells. So cells are the unit of life. So this animal has cells, full whole body has cells, and this leaf, this stem, and these roots also have cells. If we check this root in the microscope or the and electron microscope, we find many cells in this root system. If we go for root system, if we observe this root system or a single root, very thin, very small root, if we see in electron microscope, many, that means hundreds of thousands of cells will be there in this small root-like structure. So roots have cells. So stem have, so we can draw this cells. In 8th class already we learned all plants, all animals have cells in their bodies. So all these plants and animals have cells in their bodies. So we can draw this leaf with cells. Many cells will be having in this leaf. This is the green leaf. So we are drawing with green pen. So these are all cells from the beginning itself. That means when you learn biological sciences, means biology from 8th class, the first lesson or second lesson tells about these cells only. So all living organisms like plants and animals have cells in their bodies. It is filled with, their bodies are filled with like these cells. Everywhere, if we go for our brain, if we go for our, if we check our eye, if we check our all the skin, the bones, everything, intestines, heart, every part and parcel of this body contains these cells. So these are called cells, filled with cells. Our bodies are filled with cells. So in 8th class, we tell that cell is the unit of life. If there is, if we see any cell in water or on the land, then we can particularly say it is a living thing. It is from living thing. So these leaves are this leaf has many cells throughout the leaf structure. So if we observe a single cell, if we take one cell, if we separate one cell from this leaf and we if we observe what we observe in this plant cell. So in plant cell there is a big tonoplast in the center of the tonoplast, that is called vacuole. So the other name is vacuole. So the vacuole or the tonoplast is present in the center of the green cell, that is plant cell. So, and then one nucleus will be there. It will be found anywhere in cell, not in the center. So we can see the nucleus in plant cell. Some in it has 4 corners or 5 corners. So any corner the nucleus sets there, and it can be here also. If it is here, then we should rub this. So like this, that means the center of the every cell of this plant cell has tonoplast in the center of the cell, and the nucleus will be pushed towards this plasma membrane. Then the most particular important thing that plant have green color or chlorophyll because of this chloroplast. So we can say this is a chloroplast. Why we observe these leaves in green color? Why these leaves are in green color means because of this chloroplast. So chloroplasts are present in the leaves or tender parts of this plant. Then in roots, do we see this chloroplast, green chloroplast? Any root will be in brown color or black color. So we will not see any chloroplast in this root system of this plant itself. It is a plant cell only. This root belongs to this plant, but in root system there are no chloroplasts in the root system. We see chloroplasts only in the leaves and tender parts of this plant. Then first the scientists used to think that many scientists thought that chlorophyll, what we are telling, the first and foremost important factor to synthesize food in the green plants is chlorophyll. So this chlorophyll, many scientists thought that this chlorophyll will be in the whole body of the plant. So they thought the chlorophyll will be occupied in this plant body whole, in the roots, in the stem, and in the leaves, twigs, vegetables like that, fruits, flowers. All many scientists thought like that chlorophyll will be present all the whole plant. But after that, some scientists came to know that chlorophyll will not be all over the body of the plant. Chlorophyll will be present in the leaves. Chlorophyll will be present in the cells. Already we have drawn this cell. So in this part, cells are there. So to some extent we draw the cells. The whole leaf will be having cells. It is filled with cells. So each cell have chloroplasts. Maybe one, that means in leaves itself. So why I am pressing this point is in leaves, in green leaves, in each cell of these green leaves, each cell has chloroplasts, one or two or more than two, like that. It may have in some leaves or in some cells 2 chloroplasts, 4 chloroplasts, 5 chloroplasts, like that. We can observe in these cells of the leaf. So the main point is to note down the all cells which are present in the green leaves will be having this chloroplast. Because of this chloroplast, this green color is occupied in these plants. So chlorophyll is present only in cells, not all over cells of the leaves, but not all over the body of the plant. That means root system like that. We will not see these chloroplasts in stems in roots. So especially we observe these chloroplasts or these chlorophyll in the leaf cells itself. So this is the point. In leaf cells, the chlorophyll is present. And then for this photosynthesis, now photosynthesis, already we have discussed what is photosynthesis. So it is photosynthesis. First factor, sunlight. This photosynthesis, in this photosynthesis, what happens? The sugars or glucose is prepared in the cells of these leaves with the help of these four factors: chlorophyll, sunlight, carbon dioxide, water content. And these all four factors undergo photosynthesis, and they prepare sugars and glucose in their cells. So scientist Van Neal, how can we predict this photosynthesis in equation? So if we write an equation for the photosynthesis, already we know about photosynthesis. Photosynthesis means preparing food in the green leaves of green plants. And then the factors, four factors are present in the atmosphere, and then two factors are present in atmosphere and two factors are present in green plants. And the equation, how do we explain this photosynthesis? That means photosynthesis. So particularly this photosynthesis, how do we explain in equation? So first, scientist Van Neal, he is a scientist, he is a botanist first, and he made many experiments on these plants. So he gave an explanation, or one explanation that one molecule of carbon dioxide reacts with two molecules of H2O to give one molecule of carbohydrate and one molecule of H2O, that means water, and one molecule of oxygen. So this equation is proposed by this Van Neal, scientist, great scientist Van Neal. First he predicted and he gave an equation for photosynthesis. So photosynthesis in words are, if we write what is photosynthesis means, in photosynthesis sunlight is needed, first factor, chlorophyll is needed, second factor, third factor water is needed, and carbon dioxide is needed. And then with the help of these all four factors, these chlorophyll or chloroplast prepares carbohydrate and this water content, and it releases oxygen in this process of photosynthesis. So Van Neal equation is one molecule of carbon dioxide reacts with two molecules of water content, and it gives one molecule of carbohydrate, one molecule of H2O, that means water content, and one molecule of oxygen. This is the equation proposed by Van Neal. And later, and Van Neal is a great scientist, already we have discussed, and Van Neal observed this purple sulfur bacteria. So you will get an a bit in this regard. So purple sulfur bacteria. So Van Neal experimented. He has done many experiments on this sulfur bacteria that is called purple sulfur bacteria, and he came to know that sulfur is released. Sulfur, that means yes is released instead of oxygen because it has sulfur in this bacteria. So sulfur bacteria, when it undergoes the photosynthesis, this process, this purple, who gave this explanation? Van Neal, this scientist gave an explanation that in photosynthesis, when the purple sulfur bacteria undergoes photosynthesis, the sulfur will be evolved. That means it comes out, sulfur will comes out when the bacteria undergoes the photosynthesis. Then instead of oxygen means one scientist, a great scientist, after Van Neal, who gave one more explanation or who gave one more equation for the photosynthesis. This is our main process or synthesis of food content. So, photosynthesis, Robert Hill, one more scientist, he is a great scientist who worked on green plants for this to know how the green plants undergo this photosynthesis very clearly. He worked for years together to get the correct information in photosynthesis. How the photosynthesis occurs in these green plants, how they prepare their food for themselves, how the carbohydrate, that means glucose or sugar, forms in the plant trees. He worked more. Robert Hill worked more about this photosynthesis which is occurred in the green plants. So Robert Hill gave an equation. He gave an explanation. Oxygen. Van Neal gave an explanation that the sulfur bacteria, when it undergoes photosynthesis, the sulfur is evolved. That means sulfur will be come out from these plants. And Robert Hill, he worked in plants, green plants, and he worked for photosynthesis for years together. And he gave an equation 6 CO2, 6 CO2 plus 12 H2O. Van Neal gave only 6 CO2, 12 H2O gives with the help of sunlight and chlorophyll. So we came already. What is photosynthesis means? 4 factors needed. Sunlight is needed, chlorophyll is needed, carbon dioxide is needed, water is needed. Now, Robert Hill gave the explanation or a good equation which we follow in these days till today. So 6 CO2, that means 6 molecules of CO2 reacts with 12 H2O, 12 molecules of water content with the help of, in the presence of sunlight, with the help of chlorophyll. In the chlorophyll, what happens? C6, H12, H12O6 and 6 H2O, 6O. So, Robert Hill's equation. So, sometimes we get a question. What is the equation of photosynthesis? It is a direct question. What is the equation given by Robert Hill towards photosynthesis. That is another question which gives the same answer. 6 CO2 plus 12 H2O in the sunlight presence, in the presence of sunlight with the help of chlorophyll. It prepares, that means plant prepares food content. This is glucose. This is glucose. This is water. H2O means water, oxygen. So this is photosynthesis is a complex synthesis. Photosynthesis is a very complex. That means photosynthesis is a reaction which is very complex reaction. So why we use this complex, this C6H12O6, because of this C6H12O6. This is a complex material. So this is a carbohydrate which is prepared in green leaves with the help of sunlight, with the help of chlorophyll. When carbon dioxide is present in the atmosphere, when the water is present in the cells of leaves. So this equation is given by Robert Hill. And next, photosynthesis. How the photosynthesis occurs in plants and green leaves we came to know. And then if a plant prepares, what is the reality? That means how can we say that a plant, I have cut this twig. So this is from the morning itself, it is in the sunlight. So it prepares food for 4 hours or 5 hours. If it is in the sunlight, first the plant has to be alive, and then it should be in the sunlight for 4 hours and or 6 hours. After that, we have to cut a leaf and we have to test that. Already we learned that photosynthesis occurs in green plants, and green leaves will prepare food content or carbohydrate or glucose or starch in their leaves. What is the evidence? Then in science we give some evidence. So when we already discussed about this photosynthesis occurs in the green leaves itself of the plant, and in these green leaves many cells, number of cells will be there, and in each cell there are chloroplasts. In this chloroplasts the chlorophyll is present, and this chlorophyll will prepare the food content, carbohydrate or glucose in the presence of sunlight. So I have already cut this leaf after having sunlight for 4 hours or 5 hours, and then we have to test. When we say something, we should prove that sentence. And then, so I have cut this leaf, and we have to do an experiment. Already photosynthesis, what is the product? What is the product? Last product is glucose. So last product is glucose in the photosynthesis. Last product glucose. So we have to test this leaf after having sunlight for 4 hours or 6 hours, and then we have to test this leaf in the laboratory. We have in the laboratory we'll be having beakers. This is a beaker, glass beaker, and we have to put water more than half in this beaker and get ready for the experiment. And first we have taken this leaf. Already I have shown that leaf, which is under the sunlight for 4 hours, and we have to select the leaf, very large leaf like this. This leaf will not enter into the test tube. So for the experiment sake, we have took one leaf like mango leaf. It will be some thin and large like this. So we have to cut this leaf after getting the sunlight for 4 hours. So we took this leaf, and firstly we have to prepare one test tube also. One test tube. So put some methylated spirit in this. This is called test tube. There and then you have to write the name, otherwise you will forget. So this is the test tube, glass test tube. Whatever we do in physics or this botany, that means biology, the experiments, we use only glass material, glass apparatus we use in our classes or labs for observing what is happening in these apparatus. We have to see with our eyes. We have to observe. So we have already taken this test tube, glass test tube, and put some methylated spirit. Methylated spirit. Why you are doing this experiment? To test the glucose. To test the, already we have CHO combination, that means glucose or sugar or starch. Already we discussed that starch will be prepared or glucose will be prepared in the sunlight with the help of this chlorophyll throughout the in the sunlight with the help of this carbon dioxide. The leaves will prepare this glucose or sugar or starch. We are telling like that. So we have to prove also. So in proving that in photosynthesis glucose is occurred, the last product, end product is our glucose or starch. To prove that sugar is present, that leaf has undergone this photosynthesis for 4 hours, and it is in the sunlight, and it has water content. It has taken already carbon dioxide. It undergone this photosynthesis, and it has prepared this food content, that is glucose or sugars or starch. Then we have come to prove that what we have already learned. That means whether it has prepared food or not, we have to verify. So for the test, we have to take one test tube and put some methylated spirit, and then we have to take one beaker with water. This is water, and now we have to do experiment. So we have taken apparatus. Whenever we do an experiment, means we have to take this apparatus first. First, we have to take a beaker, water content, and methylated spirit, and this test tube. And we should bring after 4 hours getting sunlight this leaf. And when we do the experiment, we have to cut the leaf and put it in the this what you say, this beaker. So green leaf will be green in color. So we have to put this leaf. So methylated spirit full. We have to drench all the leaf content. We have to put in methylated spirit this green leaf, and then already prepared this test tube with methylated water, and already we have put this green leaf in this methylated spirit. Then we should do an experiment. Now, experiment is we should take one tripod stand and one Bunsen burner. So tripod stand asbestos net will be there, and on this tripod stand we have to put this baker. Already we have prepared this baker with water content, and then what we have already prepared here, this apparatus called test tube with the green leaf and with the methylated spirit, we have to put this test tube slowly in this water. So put the test tube with leaf. Methylated spirit is there. So we can take so this is the experiment. So we should show methylated spirit in this test tube and the leaf, long leaf having this long leaf in the methylated spirit. So this is the apparatus already we have prepared for the test. That means what we are testing now. So food, that is the end product of photosynthesis. What is the end product of the photosynthesis? That means what these green plants or green leaves have prepared in the sunlight with the help of carbon dioxide, with the help of water, with the help of uh, this water content and sunlight, chlorophyll. So the end product we are testing the carbohydrate CHO or C6H12O6, that is the end product. We call it glucose, and this glucose will be turned into starch. Afterwards, it will turn into starch. First, in glucose condition, it will be prepared in the leaves while they undergo this photosynthesis. So we are testing this carbohydrate or sugars, glucose we can say sugars also. So glucose or sugars we are testing now whether the leaf has prepared in the photosynthesis, whether the leaf has prepared this food content called glucose or sugars while undergoing this photosynthesis in the presence of sunlight. So we have to lit this Bunsen burner. We have to put the lit this Bunsen burner. After 20 minutes or 30 minutes, the water will be boiled, and the methylated spirit will be get heated, and the leaf will be, what happens, turns some pale in color, and then we have to take off. After that, after having that means 20 minutes to 30 minutes we have to do like that, and after boiling the water for 20 minutes we have to take off slowly this test tube outside of the apparatus. So test tube we should take out after boiling for 20 minutes or 15 minutes like that. Methylated spirit is there, and the leaf also is there. We have taken this leaf from leaf with this, what you call this methylated spirit. So full, the leaf should be full in this spirit, not half or a little bit. We should not leave this leaf part outside of the this methylated spirit. So fully drenched leaf. That means this leaf should be drenched in the methylated spirit, and then we have to take off that leaf outside, and this leaf we have to test with iodine iodine solution. So take iodine solution, and after that you have to pour in this. You have to put iodine solution on this leaf, and it will urgently. That means it turns into blue in color. This green will go off. Green color will be vanished from the leaf, and the leaf will be dark in blue color. That means if the leaf turns into blue in color with the help of this iodine solution, then we can definitely say the starch or glucose is prepared. So we can test this leaf before putting this leaf in the sunlight also. That means early in the morning, if we get up early in the morning, 5 o'clock to 6 o'clock, then we have to cut one leaf. So we have to tally those two leaves also. We can tally. So early in the morning we have taken one leaf. We have already cut one leaf and put it aside in the dark room, and then one leaf after 4 hours again. That means after getting sunlight for 4 hours, the other leaf. Then this other leaf we have to test like this, and we have to do the experiment like this, and we have to test with iodine solution. Already, if we get any doubt, so this leaf will be turned into glucose. Then already we have taken in the early in the morning, 5 o'clock, we have taken and put in the dark room. We also bring this leaf, another leaf, and if we test that leaf, it will not turn into blue. So another leaf we have already cut in the early morning and put in the dark room. So, with these both leaves, we can do experiment easily. Then we can observe what happened with the sunlight. So, like this also we can do this same experiment with two leaves. That means before getting sunlight, this leaf is before getting sunlight, means early in the morning we have already cut this leaf and put in the dark room, and this leaf is getting sunlight for 4 hours or 5 hours, and we have to cut this leaf and put in methylated spirit, and this apparatus should be put in the water of the speaker, and Bunsen burner will boil this water, and with the help of this Bunsen burner, then we have to take after 20 minutes or 30 minutes this leaf outside of the apparatus, and we have to test with this iodine solution. These two, this leaf is before getting, that means before getting sunlight, before sunlight, that means dawn, in the early morning. This is after getting for sunlight for 4 hours or 5 hours. So what is the difference? We have done the experiment with the iodine solution, which is which has not get this sunlight, and we have done the experiment. That means we have put iodine solution on this leaf which got sunlight for 4 hours. This leaf does not get any sunlight, so it will not turn blue in color with the iodine solution, whereas this leaf after getting 4 hours sunlight it will prepare definitely food that is called glucose or sugars which turns that iodine has the character, it turns any starch that is glucose into blue in color. That is the main important thing we have to identify. Iodine character is turning. If we take a wheat flour or rice flour in some glass, that means watch glass, and then on that flour, wheat flour, you put two pinches of wheat flour in the petri dish or watch glass. Then you have to put iodine solution. Then the flour suddenly changes into blue in color because it is a starch, because the wheat flour or rice flour are the starches, so it turns into blue in color when we put iodine solution. So this leaf does not turn into blue in color. Why because this leaf has not get any sunlight, because we have already cut this leaf before the sunlight, and already we put in the dark room. So this leaf is lucky to get sunlight for 4 hours, and after that we are doing this experiment. If we conduct this experiment perfectly, then we get the good result. So this is the lesson we have already discussed. So we can meet in next class. Thank you.